A shows HeLa P57 cells in the HeLa CTRL cells after being treated with doxycycline. Doxycycline is used to treat infections, so it's seeing the results after the cells are treated. Part B of this figure shows the results 24 hours with the doxycycline, and they were immunoblotted after those 24 hours, and the results show that the cells were still visible and still continued to grow. Figure 2 shows the HEK293 cells after they were injected with the immunotag infection. This was looking to see if the cells would clump together after those 24 hours. Then, after those 24 hours, they were immunoblotted. The cells were then immunoprecipitated. In part B of this figure shows the HeLa cells that were transfected with the expression vector. Part B of figure 2 shows the HeLa cells that were transfected with the expression vector. After the transfection took place, the cell abstracts were immunoblotted. The total acts were then immunoprecipitated. In figure 3, we are looking at the 57-KIP2 in the LIM kinase and the LIMK1 that is colonized in the cytoplasm. In figure 3, we are looking at the P57-KIP2 and the IMK1 that is colonized in the cytoplasm. In figure 3, we are looking at the P57-KIP2 and the LIMK that is colonized in the cytoplasm. Figure A and B, the HeLa cells were transfected with the expression vector, and they were transinfected for 24 hours. This figure shows the fixed and stained cells. Eyes were specifically used to see if the MYC immunotagged LIMK1 was present. The nuclei were also stained just to be able to differentiate from the cells. The yellow pigmentation is a result of the overlay between the red-stained P57-KIP2 and the green-stained LIMK1. This overlap determines the colonization between proteins. In figure B, we can see the results of the cytoplasmic and nuclear extract that were immunoblotted. So overall, figure 4 shows the promotion of the LIA1. Part A depicts the HeLa cells that were transfected with the expression vector. The expression vector was then transfected for 24 hours, and we can see that in lanes 5 and 6 were treated with a chemical inhibitor for 3 hours before the protein extraction. The extracting protein cells were then analyzed using immunoblotting. Standard for equal loading of the proteins, GG3PHD, was used. Part B of this figure shows the HeLa cells that were also transinfected using the expression vectors for coding. They were transfected for 24 hours, and then after the 24 hours, the total cell extractions were then immunoprecipitated to be able to measure the kinase activity of the LINK1. ATP was incorporated into the coflin. The amount of the LIMK1 normalizes the activity of the subject in the reaction. Part C of the figure depicts the HeLa cells being transfected with the expression vector in 24 hours. After the 24 hours, they were immunoblotted, and the protein cells were extracted and analyzed by immunoblotting. After the 24 hours, the total protein cells were analyzed and immunoblotted using multiple different antibodies. As a standard, G3PDH was used for equal loading of the protein. For part D of this figure, the HeLa P57 cells were transfected with the MYC LIMK1 or the MYC LMIK1 D460A, and they were treated or kept as a control with the doxycycline for 24 hours. After being treated, nuclei were counterstained. This allows us to see the images of the immunostaining.
The last part of figure 6 is going to be part C. And this figure shows the confocal imaging of the immunostaining with the protein that has the CDK inhibitor. This protein, the P57, is seen in green on this immunostaining. The F-actin is in the red, and the nuclear hoax staining is in the blue. And once again, kind of the process of this was the HeLa cells were transfected with this protein and the CDK inhibitor. And then kind of in the fourth panel of this figure, you can see that they're all merged together. and it's kind of just a combination of all of those staining. So the first part of this figure, part A, we can kind of see the quantitative results on this graph here, and this is kind of showing the fluorescence recovery after the photo bleaching. So each plot on this graph is representing the recovery after the bleaching, and there's kind of times listed at the bottom. So we go 20, 40, 60, 80, 100, 120, up to 140, and this is time in seconds. So kind of this process, the HeLa cells and with the protein were transfected with the GFP actin and treated with doxycycline, which is noted up there as DOX, DOX. This next part, part B, shows those confocal images of the untreated and DOX-treated cells. Doxycycline treated, which is noted as DOX on this photo bleaching. This shows the recovery of the GFP actin fluorescence after the photo bleaching, and this is measured in milliseconds. So the first part of this figure, part A, is showing those contrast phase images at different points after the wounding. So we've got 0, 1.5, 3, 4.5, and 6 hours of time here. The controls showed at the top panel, and then the cells that were treated with the doxycycline or DOX are at the bottom. This is kind of showing the progression of cells moving into the wound area. Those HeLa cells with the protein cells, they were untreated, which is the control up at the top, or treated with doxycycline, which is the panels at the bottom. This next part of the figure, part B, also utilizes those HeLa cells with the protein cells, and they're either untreated, the control, which is noted as kind of that light gray here on this, or treated with doxycycline, which is noted as DOX, DOX, which is the black. Shown are kind of the measures of distance between the edges of the wound after kind of relating to that same progress of cells that's moving into the wound area. And we can kind of see that there's differences in those kind of height of bars. Finally, the last part of this figure is going to be part C. This shows a schematic model of the effects of that protein on actin. And it's showing in the absence of the LAMK1 in that protein, the colfilin depolymerizes that actin filaments, and when the protein is expressed, it is translocated to the cytoplasm, where it interacts with that LAMK1. This interaction between that LAMK1 makes it more active by means of that phosphorylating colfilin, which makes the colfilin unable to depolymerize that actin filament. And this process is kind of showed in that schematic model, just explaining this whole process.